Every human body operates as a staggering biological ecosystem of 30 trillion individual cells. For an organism to exist, these microscopic units must flawlessly duplicate 3 billion base pairs of DNA and divide with unflinching precision. This selfless cellular coordination allows the body to develop from a single egg, specialize into neural networks, and heal from traumatic injury. Individual cells communicate across vast biological distances, dividing only when instructed by systemic hormones or local growth factors. For hundreds of millions of years, life on Earth existed solely as single-celled organisms, driven by the innate imperative to replicate endlessly. The transition to complex multicellularity required a difficult biological paradigm shift. Individual cells surrendered their independence to ensure the structural integrity of the collective. That harmony is perpetually fragile. When acquired genetic damage strips away a cell's cooperative programming, a microscopic vanguard stops following the rules. A single rogue entity reverts to an ancient imperative, ignoring biochemical signals to stop dividing, and instead siphoning off the body's nutrient supply. The biological engines responsible for cellular growth and healing, the very processes that sustain human life, are the same engines that make cancer an inherent vulnerability. Public perception often frames this disease as a uniquely modern affliction, a toxic byproduct of synthetic chemicals and industrialization. However, the historical record reveals that cancer has plagued complex life since human antiquity. The oldest known surgical treatise, the Edwin Smith Papyrus from ancient Egypt, contains the earliest written descriptions of aggressive breast tumors. Egyptian physicians recorded treatments like cauterization, but for advanced tumors, they made a chilling observation. They were completely untreatable. Centuries later, Hippocrates observed advanced tumors radiating outward like a crab's limbs. He named the disease carcinoma, establishing the root of modern oncology. While early theories of humoral imbalances were incorrect, these rigorous observations shifted the perception of cancer. Categorizing it as a devastating biological process rather than a divine curse was the first step toward a scientific solution. To understand the cellular rebellion, we have to look at the microscopic molecular control systems that usually prevent it. A healthy cell is governed by the cell cycle, a sequence guarded by rigid biochemical checkpoints that act as molecular gatekeepers. Central to this defense is the tumor suppressor protein p53, known as the guardian of the genome. When a cell suffers genetic injury, p53 halts the division cycle, granting the cell's repair systems time to fix the broken code. Or, if the damage is too catastrophic, initiating a programmed cellular suicide called apoptosis. Cancer develops precisely when these fail-safes experience catastrophic failure. In the vast majority of human cases, the TP53 gene is deleted or heavily mutated, causing the critical molecular breaks to fail. Without a functional p53 pathway, cells harboring severe DNA damage are permitted to divide, completely bypassing natural safeguards. These rogue cells accumulate ever-increasing arrays of mutations, becoming progressively more unstable and resistant to external growth signals. This is the foundation of the rebellion. The cell fundamentally abandons its cooperative programming, devolving into an ancient, highly resilient state of autonomous, unconstrained growth. This microscopic failure translates into a staggering global burden that defines the public health challenges of the 21st century. This data reveals the sheer scale of the crisis, with lung and breast cancers leading globally in both incidence and mortality. As populations age and undergo millions of cumulative division cycles, the statistical probability of genetic replication errors virtually guarantees the accumulation of damage. The modern scientific fight was initiated by the National Cancer Act of 1971. This legislation shifted oncology from a purely surgical discipline into a molecular science, funding the discovery of the first human oncogenes. Before this molecular revolution, treatments were largely blunt instruments. Early chemotherapies, derived from chemical warfare agents like nitrogen mustard, indiscriminately attacked all rapidly dividing cells, causing severe collateral damage to healthy tissue. The clinical challenge is rooted in biological mimicry. Because cancer cells are built from the host's own molecular blocks, they wear an immunological uniform that allows them to camouflage themselves from the body's natural defenses. The Hallmarks of Cancer framework explains how tumors aggressively adapt. They acquire new traits like phenotypic plasticity to resist pharmaceuticals, trigger angiogenesis to feed the mass, and manipulate senescent cells to secrete growth factors for the surrounding malignancy. A tumor functions as a highly heterogeneous, rapidly evolving parasitic organ that requires ultra-precision molecular logic to successfully dismantle. To find a solution, modern science looks beyond human pathology to the grand tapestry of evolutionary biology. Statistically, organisms with more cells and longer lifespans should face a proportional increase in cancer risk. A blue whale with 1,000 times more cells than a human should face a mathematical certainty of developing the disease early in its century-long life. This is Peto's paradox. Despite their size, massive cetaceans rarely, if ever, develop clinical malignancies. Genomic sequencing of these deep-sea giants reveals that they have evolved redundant tumor suppressor genes and highly elevated levels of DNA repair proteins. Their biology has adapted to suppress cancer thousands of times more effectively than the human genome currently can. This evolutionary triumph proves that biological mechanisms capable of suppressing cancer already exist. The goal of modern medicine is to decode these natural defenses and replicate them pharmacologically. In 2026, human oncology is moving entirely away from broad cytotoxic approaches towards bespoke molecular precision. Antibody-drug conjugates act as molecular smart bombs. An antibody component navigates the bloodstream until it selectively identifies a target antigen on a cancer cell, whereupon it internalizes and releases a toxic payload exclusively inside the malignancy. Simultaneously, immunotherapy focuses on stripping away the tumor's molecular invisibility cloak. Checkpoint inhibitors block the chemical brakes that cancer hijack, unleashing the full power of the host's T cells to destroy the mass naturally. Artificial intelligence now interprets multimodal data from liquid biopsies, identifying minuscule fragments of circulating tumor DNA years before physical symptoms manifest. Cancer remains an inescapable vulnerability embedded in the architecture of complex life. But by reinforcing the biological boundaries that keep our cellular society intact, humanity is finally learning to outsmart its own biology.